The fact that someone has cancer means the system has failed.
What is a CAR-T therapy?
You can take the already existing cells, modify them and give them back to actually treat the patient.
You take them out of someone's body, you stick a receptor on them to go and kill the cancer in your body.
Emily Whitehead is the fourth human ever to get a CAR T-cell therapy.
Science is essentially moving slower than your patient needs.
Science, in fact, does move very fast, but still we have the big patient need and we still have a lot of tumor entities where we have no cell therapy.
If you have the perfect solution but you can't distribute it to patients, you don't have a solution at all. Hello and welcome everyone back to the DeSci Podcast. I am your host Ella and I'm joined today by Henry, who's going to be talking to us about the interesting topic of immunotherapies and CAR T-cell therapies, which I think probably sound a little bit obscure or complicated, but that will be Henry's job today, is to explain a little bit more about us and sort of maybe the context of how Henry has ended up on the DeSci Podcast is he is looking to start his own project and will be going through the molecule IP protocol pipeline, and so has approached us and we've been chatting, and I thought it would be a great time to use it as a learning exercise to teach more people about immunotherapies. So yeah, Henry, we'd love to know a little bit more about you.
Yes, nice of you to have me. And I'm Henry. I'm a medical doctor, freshly approved at the Charité Berlin, and I always had a strong interest in cancer therapy and children's oncology, and then heard about the so-called CAR T-cell therapy, a mode of adoptive immunotherapy, a cell therapy that we're going to talk about, and someone then got into the loop, did my doctoral thesis on that, and now propose a project that should increase efficiency and safety of the system if it works out quite well.
Amazing. So I think it would be helpful, to take us from the top. What is a CAR T therapy?
Yes. So in general, CAR T-cell therapy is a special type of what we call adoptive immunotherapy, and this is in general a therapy where a patient, normally cancer patient, but sometimes autoimmune patient, gets a set of immune cells that have been derived either from himself or a different person. So maybe the first instance of what you could call adoptive immunotherapy would be stem cell transplantation, which is kind of a wild ride. Who gets it is we say third in cancer medicine in there. It's like a third of patients is cured, a third of patients eventually gets a relapse, but a third of patients can even die from this type of therapy. Wow. So this was kind of the old last resort method, the allogenic stem cell transplantation. And then there is something that is not that much invasive, the autologous stem cell transplantation, where a patient gets his own cells harvested, then gets intense ablative chemotherapy to get rid of all his stem cells, and then gets the collected stem cells, and often they're kind of purified to single out them against cancer cells and gets them back in the hope of remission. And CAR T-cell therapy kind of is the next level of this, where we try to combine the advantages of both without posing that much danger to a patient. So in generally, we again collect, this time not stem cells, but T-cells normally, also possible with NK cells, immune cells from a patient through...
Can you just tell us what is a T-cell?
Yes, a T-cell is a type of immune effector cell. It's a type of white blood cell that comes in two categories, the CD4 and the CD8 T cells. And one of them is possible to recognize diseased cells, whether they be virus infected or be tumor cells, and alert other cells. And the other type, the so-called CD8, more interesting for us, is able to recognize these cells once activated and then kills them via a specific receptor or by throwing a cocktail of toxic proteins at them. And the NK cells are kind of doing the same job as these killer T cells. So the NK means natural killer cells, but they are like kind of your ground force police. They are not that specialized as the T cells. T cells normally come with one specific receptor for only one type of antigen, and the NK cells they have different kind of pattern recognition receptors, and if something looks too much off, they also can kill a cell by throwing toxic peptide on it.
Interesting.
Yeah.
I mean, I think like when one thinks of cancer therapies and stuff, one tends to think of like chemotherapy and radiotherapy. And so I like in my mind, and I don't know if this is a correct perception, but that cell therapies are kind of like a little bit more like left wing, a little bit more expensive, boutique, and kind of higher risk but potentially higher reward. Would you think that's a fair assumption?
In generally, yes, there still is difference among them. So there are attempts to use cell lines for this, which can cut down costs. What is a big cost driver is producing them individually for a patient. So having to collect patient material and then genetically engineer, like with the approved CAR-T cells that are by now, with a receptor that is specific for every single type of tumor target and every single antigen that has different kind of receptors and needs to be done individually, that can get it rather expensive by now. So CAR-T cell therapy, so basically to get back to it, it is like we take these T and NK cells and put an artificial receptor on them, a so-called chimeric antigen receptor, which is part of an antibody and intracellular stimulatory domain stuff that can activate the cell to kill, and then it's given back to the patient. And the big cost driver is we have to take apheresis from the patient. So we have to collect the cells from the patient, then culture them for a few weeks, transduce them, so put this receptor on them, and then give them back.
Okay. And then those, so basically like if I bring it back down to like, I think a very simple language is you'll take these sort of immune cells that would normally like kill maybe infections in your body or that sort of thing. You take them out of someone's body, you stick a receptor on them, let that grow up and culture it. And so and then you give it back to the patient. And by adding on this receptor, you've now given those killer cells the ability to go and kill the cancer in your body.
Yes, indeed. So actually, these are also already the cells that normally kill cancer in anybody. So the NK cells are actually the specialized cells to kill any aberrant cells which might be cancer. But the fact that someone has cancer means the system has failed and the tumor cells were able to escape the surveillance. And this is a mode we employ to restore this surveillance so that the immune system can recognize the tumor again and we can kill it off.
Interesting. I think it sounds like quite an intuitive solution that sort of you just are restoring the body's natural system to do what it was sort of originally meant to do.
Yes. At the first thought, then we have to take several shortcuts because sadly most cancer cells don't have anything that is completely mutated and distinguish them completely from everything that is healthy, because those cancer cells would have been killed off. They would never have grown out. So CAR-T cell therapy as it is used now is we have receptors for antigens, molecules that are on cell surfaces, which are like on expandable tissues. So what is approved now is against an antigen called CD19 or an antigen called BCMA. They are both on different B-cell malignancies. And even if it comes to the fact where all these cells that have this are killed off, you can live without them. So some patients have to substitute immunoglobulins, but still they're cancer-free. So,
what is an immunoglobulin?
Antibodies. Actually,
Okay.
this is we probably all have heard of antibodies during COVID more than enough. So as these B cells is also a type of white blood cell that produces these antibodies to fend off
your
infections.
body's defense system.
Yes.
Essentially.
And these B cell malignancies are actually mutated B cells. They once were B cells or B cell precursors, and then they just grow out and grow into blood cancer. And this is, so by killing off the B cells, we also kill them off, and then you have to do the job of the B cells in some cases, not in all cases, externally.
Yeah. So I mean, maybe just like I'm not sure if you know these numbers on hand, but like what percentage of cancers are good candidate, like cancer types, and maybe also cancer patients are would be good applicants for this type of therapy?
This is really different between how you construct your actual cell to use it. So what is approved for B-cell malignancies, so blood cancers. On the one hand, blood cancer is easier to target with this type of therapy because it is more accessible for the immune cells. The solid tumors always have very immunotoxic, toxic to immune cells, microenvironment, and tend to regulate them down. There is approaches though to tackle this, but it's kind of more difficult, and it's still at the beginning of building this up. What is approved is good for B-cell blood cancer types. So that's the multiple myeloma, already approved, diffuse large cell B-cell lymphoma, the ALL, acute leukocytic leukemia, which also was the initial patients where this was trialed. But in general, you could tune it to tackle any type of cancer. Thing is, you need a good target, a target that can be targeted safe, also where we want to jump in with our project. And you need to get to the tumor cells with the still active immune cells.
Yes. Okay. Interesting. And I think, one of the things that pops up into my mind immediately is for an immunotherapy or something that you sort of are adding in immune cells that have been sort of modified. My first thought is there also quite a high risk of it completely, sending your body into like a tailspin of like an immune cascade? And what is the risk level for something like that?
It does happen. It is actually quite a regular thing. So this is also the cause for this to be a therapy which has to be induced in the hospital setting. So the transfusions always happen in the cancer center with at least a few weeks of presence there to surveil the patient. What is the most common big adverse event is cytokine release syndrome. This is where inflammatory mediators from the hyperactive CAR-T cells, as they all then find the big tumor load and start to killing this off, are just flooding your blood and systems, and you get like a graft versus host like disease, where the immune cells start attacking around. You can get high fever and just general runaway inflammation.
Yeah.
What we do is we give an antibody called tocilizumab, anti-IL-6. This is the main inflammatory marker that is elevated in this, and with this antibody we can tune it down a bit, and then it's kind of we have to wait if it happens. So some patients never get it again after the big tumor load is gone. Some patients have problems with this all along. So in some cases there has to be an abortion of these therapy where then the cells have to be killed off. So modern CAR-T cells have a so-called suicide switch. So if you give a specific drug, they are genetically engineered to die then.
That is so interesting to me that sort of you're plugging in these suicide switches. I think it's quite clever to have this like emergency shoot.
It's an essential safety feature, yes. Yes, indeed.
Interesting. And I would like to know, like, why, I mean, obviously this is something that you are dedicating your life to, you know, building these therapies. What was it that drew you into this topic so much?
Actually, I've heard about this quite early. So it was a high school presentation I did on broad level of genetic engineering in medicine. And then I found out it was kind of a new topic then, first in-patient trials, which were performing quite well. And yeah, I just found this idea extremely cool that you can take the already existing cells and then modify them and give them back to actually treat the patient, which otherwise often would be terminal. So I read about the original story of Emily Whitehead, is like the fourth human ever to get a CAR-T cell therapy, and the first to make her story public. So she at that time she was a girl of 7 years. So in 2010, at the age of 5, she was diagnosed with acute lymphoblastic leukemia and was initially treated with a normal regimen, then had several relapses, and in 2012 it was like getting pretty grim for her. So doctors started telling the parents, well, plan your last things you want to do with your daughter. And then they enrolled her into the phase 1 initial study of what today is known as Kymriah by Kite Pharma, Tisagen Lecloitzil, one of these CAR-T cell therapies against CD19. And yes, she actually, when I read about it, she was 3 years cancer-free and is to this day.
Wow.
So it was like, of course, an extreme success story. But if you read like from, okay, she's as good as that, to 22-year-old woman with all her life ahead of her, this was amazing. So yeah, later on when I started studying medicine, I always had this at the back of my mind, and then looked out for a group where I could learn more and work with this myself. And then yeah, I found Armin Reem's group at the Max Delbrück Center here in Berlin, who worked a bit with the CAR-T cells, and I did experiments on them, co-express them with chemokines to improve invasion into the tumor niche. As we said, like the invasion into the tumor niche was one of the big problems. So, and yeah, then I came around this general problem we have with finding a good target, because this is kind of very, very hard. We need to find something that is expressed in most patients, ideally all, but at least most, but still can be killed off without really harming the patient too badly. And this is very hard to find. So something that's completely new, completely mutated through, this is something you would have to design individually on one patient. Like not only have a CAR engineered patient individually, but really have it designed for every patient, and this is like, that's not possible. You can't do this. It would cost several millions. So that is not an option. Then we have what is approved and what works quite well. We have the CD19 CARs, we have the BCMA CARs, which have generic antigens that are expendable. You can live without these cells. And it is good for the patients which can profit from this. But on the other hand, they also have drawbacks. So, for example, the BCMA CARs available for treating multiple myeloma are not as good as like treating an ALL with the CD19 CAR. So a story like Emily's has still to unfold there. After a few years, they will relapse.
Yeah.
So
okay, interesting. So that's
still,
also, there's some of them that only sort of work for a certain amount of time, and then presumably do they have to do the whole process again?
This is the problem. Once you have done it, you could just change the antigen, but there's nothing on the market by now. So this is kind of Sorry,
I'm interrupting. Yeah,
it is still very good for the patient, as like it buys you 4 more years at the end, maybe, but it won't get you rid of this tumor of this type.
Okay. Yeah, so what I was saying was that like science is essentially moving slower than your patient needs.
Science, in fact, does move very fast. In the last 10 years, all this came up in the last 10 years. There was nothing before that, but still, we have the big patient need, and we still have a lot of tumor entities where we have few or nothing, or at least no cell therapy. And this drove me to work in this field.
Yeah. I think maybe then this is a good time to maybe tell me a little bit more about your project and what you're working on.
Yes, so we are GoCart, and I will allow myself to share screen if you say yes.
Yes, of course.
And well, we are GoCart, and we are our DeSci project that aspires to be a startup in this field. And what we develop is the so-called AND gate. So the problem I've already talked about is like the CAR-T cell has its receptor and kills off like any cell that has this antigen. But these cells will all die, whether this be a tumor cell or a healthy cell. And for these BCMA CARs, this is actually the third best option at best. Much better would be something called CD56, for example. Problem with this, it is also expressed on like every one of your neurons, of your nerve cells, of your brain cells.
Okay, we cannot kill off every brain cell.
Killing them all off is kind of counterproductive. at least that's my opinion on this one.
I would have to agree.
Yeah. So, but as it is such a great target, and we have lots of targets which are like this one, we need a novel system for this if we want to attack them. So the idea is the so-called AND gate, where we've combined 2 different targets. What we want is have like this kind of Boolean table, where we have a healthy cell that has no antigens for these cells, so no surface marker. And then we have healthy cells that have maybe CD56, or healthy cells that have the other one. You could use CD20 or CD10. And we have the multiple myeloma that has both. And if we now can tune our cells to only kill those cells that have both of these targets, then we can selectively kill off the tumor and your neurons are safe. So you don't lose your brain, in fact. Yeah, there has been ideas and trials to do this. So originally, just slapping on 2 different CARs on the same cell is rather easy. But this is the so-called OR gate, so it will kill off all cells that have one of the targets as well.
Which
so
is doubly as bad.
Not necessarily. You can still use this as a safety mechanism. So if the tumor has 2 antigens, and then we attack both, it is more difficult for him to downregulate both and still live than if you had only one. So you can use it in this way, but you can't use this for CD56. Then we had the so-called SynOc CARs, where the first CAR is not really a CAR, but a receptor that triggers expression of the second one. So the first one binds and activates the production of the second receptor. And then only this second receptor will kill off a cell. It is better, but if you think enough about it, you maybe come to it, so it is leaky. As soon as the second receptor is produced, this cell can move wherever and still find your brain cells. So
interesting.
yeah, it still is not safe enough to use it for this kind of antigen. The same goes for so-called split CARs. So CARs have inside the cells, they have different stimulatory domains, and you can split them up and distribute them on different CARs. But then the same problem, even if only one of them binds, the killing will be weaker, but there will still be killing. So same problem with them. And then we have the first approach are so-called link CAR-T cells. Another split CAR, but this one uses downstream activation domains intracellularly. So maybe to explain this, in the cell you normally have a signaling cascade. So if a receptor is activated, it activates a protein inside the cell that activates other proteins and so on, and you have a chain reaction that in the end extends the signal and gets a really high signal for the cell to do stuff in the end. And those link CARs use not the normal receptor domains, but they have downstream domains of a kind that always have to go together. So this really is a CAR that kills only cells that have both antigens. So problem solved. Not that much, but a big step. So you use downstream domains there. That means all these signal enhancement you have by having the receptor activate a protein that activates a second one and a third one, you miss out on a lot of those. So these ones still have problems with low levels of antigen expression, as tumors like to react to therapy by doing this. So big step into the right direction, but probably not the ideal solution.
Okay.
And then we come to our own approach. So we at GoCart want to push this whole AND gate outside of the cell. So we have one generic trunk for the CAR-T cell with one receptor. That is the same receptor for whatever target you want to target. And you target your antigens by so-called specific AND gate binders. They are like a binding protein for these surface markers. And on the other end, they have what we develop our proprietary binding domains that can dimerize. So if you have only one antigen on the cell, one of these binders will bind and stay as monomer.
Can you just could you explain that for us in sort of maybe some like very, very simple terms?
Yeah, so actually what we develop is kind of an adapter. So we have kind of the same CAR, the same receptor on the cell for whatever, and then the patient gets adapter proteins from us, which bind to the different antigens. So CD56 and CD20, for example, for the multiple myeloma. And only if those are together on the same cell, then these adapters can bind together. They stick together when they're on the same cell, and only when when they are joined, when we have both of them, then our CAR-T cell can bind to this and will kill the cell off.
Okay.
Yeah.
So it's really refining how your CAR-T cell is going to bind and kill.
Yes, indeed.
Okay. And so what was it that brought you to sort of decentralized science? Why do you feel like this project is a good match?
Yes, actually, the reason is we are rather early, and still this needs a number of initial funding, and should have a decent pace at which we have to operate. And also I really like the idea of like community owning the IP and community governing our decision. So this is also relevant for us as we want to open up development of these binders and are looking for collaborators on that. So we are all also open for ideas from the community for new targets. So as we say, initially we want to go for CD56, as I'm already talking about this all the way, and CD133 and another one CD20 or CD10. But we want a suite that is as big as possible. So in the ideal future, you have a doctor that phenotypes the tumor from a patient, and then he orders our CAR-T cells. And as we have the same vector, we can streamline the process. We produce the same CAR-T cells for whatever tumor it is, whatever indication. We have one process for the cells, which we hope to drive down costs by doing this. And the doctor orders the set of binders which fit the tumor expression pattern of the patient, and he can even customize this. So after the patient got the cells, he then can swap them out if needed. If the patient gets resistant, phenotype again, look what the tumor expresses now, and maybe you find another binder from our suite to get there. And all this just makes sense to me to do this in a more public way, to open this up for people to contribute to it and give us their ideas and also profit from our development in the end.
Yeah, amazing. I'm so curious, how did you discover decentralized science? Like, what was your first entry?
This is actually funny. So I met one of you molecule guys at a conference, in fact, and all the way along I was interested in crypto, found it an interesting concept, and then I heard, okay, we finance research with crypto. We do like crypto patent shares, if you want to call them so IP-NFT tokens. And this this intrigued me, and then we had a nice talk, and found that was actually kind of a good fit and a nice opportunity to build this out in public.
Amazing. Cool. I think there's one thing that's still like nagging on the back of my head, which I wanted to ask you about is, I think CAR-T. It's somewhat of a deep, like a precision healthcare, and so I want to know: do you think that the future of medicine is precision healthcare?
Definitely, if we can bring the costs down. So this is actually also the idea we want to pursue with our modular system. We need to be precise in what we do, but it needs to be modular. We can't have a big load of different cars or different interventions, which are all done patient individually. It would be great if the world had the resources to deliver this to everyone, but we need to have a point where we can standardize things. And I think modular approaches can be the solution there that brings individualized therapy to the broader public than it is now, also worldwide.
Amazing. Yeah, I think that makes a lot of sense. I think there's just like simply not enough people, resources, scientists, hours to be doing tailored healthcare to every single person. But if you just have these sort of Lego blocks that you can sort of build unique houses to each person's circumstance, I think that makes a lot of sense to me.
Indeed.
Amazing. Cool. For our listeners out there, how should they get in contact with you if they're interested?
Yeah, so our corporate LinkedIn and our corporate X is already online. And we are right now ramping up to get the Telegram channel running. So if you're really interested in the project or the to-be token, which we plan to launch like in the week after DeSci Berlin. So the date is June the 16th. Then I would advise you guys to hit us up on X or follow our LinkedIn and get the updates on our proceedings.
Amazing, yeah. And I like that you also were sort of making a call for people to contribute their ideas and that sort of thing as well, because I think that's what's quite lovely about DeSci in general that I've seen is that there's a lot of different avenues to contribute to a project, whether that be some marketing skills or, knowing the right suppliers that can, and, helping facilitate that, or having ideas, or of course, funding. There are lots of ways to help. Yeah, I was going to say there are lots of ways to help progress science. And I think I think that's such an appealing draw of DeSci.
Definitely.
Amazing.
So.
Cool. Well, thank you so much, Henry, for giving us a sort of a deep dive into CAR-T therapies and sort of these potential, sort of this new avenue. I suppose maybe I shouldn't say new, but it's running on about 10, 15 years. But still actually, relatively new.
Yeah, relative. Well, this is also kind of like research and approval are different pace. So the first ever published CAR in a lab was built in 1987. So this is the first instance of a receptor composed of a T-cell receptor with antibody parts, is what it was called, by Yoshiki Kuwana at the time. And around year 2000, it slowly got into planning of clinical trials. And then, Emily Whitehead was one of the first patients to get it in 2012.
2012, yeah. Quite crazy that it's how many years is that? Almost over 25 years. Is it? Is my math correct? Yeah. To go from lab to patient. That feels a bit scary, actually, that it takes so long for something to reach a patient.
Yeah, well, this is actually also the thing with translation. So it takes some time to get it out of the lab. Also, something I see as my background, I'm a medical doctor. I want every kind of treatment immediately because I see the people. But then you have to get it approved, which is kind of a big regulatory thing to undertake and also very costly. And then you need someone to pursue this, and this is maybe the bottleneck where things get out of university and then get into startups as this. So this is also why we want to build it as a startup in the first place. So we hope to accelerate deployment in this structure that we are building, and by using DeSci.
100%. I think it's always great when you get the sort of phenotype of someone who's a scientist, but also has business proclivities of okay, cool. I think in my experience when I studied biochemistry, and there are a lot of people who want to come up with the perfect solution, without really ever conceptualizing how that gets implemented. And if you have the perfect solution but you can't distribute it to patients, you don't have a solution at all.
Yes, actually. And this basically, yeah, the sad thing about this is often like these projects then end up in some drawer because you don't find someone who really understands what has been built and is willing to spin out, because like in the academic setting most people have their commitment over there and then move on to the next project and do not want to like dedicate 20 years to further developing it. So then it's patent then sold off or licensed out or something like this. But of course it's not the original people who conceptualized it to apply it then, and this also then takes a bit more time.
Interesting. Yeah. I think, in DeSci we speak so often about like all of these sort of breakage points where you can just have this leak out of like really good ideas and talent and that sort of thing. So I think I'm glad that you are someone who is working on those things.
It's
cool. Amazing. It was so good to chat to you, Henry. And yeah, you heard it. Go give the GoCart page a follow. Stay up to date with what they're doing. I'm really excited to see what comes next.
Yes. And
cool.
Also awesome of you to have me, and shout out to my wonderful team who helped me build this.
Yeah, always a team effort. Cool. Amazing. Have a beautiful day, Henry.
Okay. You too.
Bye.
Bye-bye.